Wonderful crowd in person for the first grand rounds of the academic year. And I see there are many, many more people online. And at the end, we'll take questions both in person and online. And I'm really honored that for this first speaker of the academic year, we have Ethan Dmitrovsky, who's director of the Frederick National Laboratory for Cancer Research, which works to combat cancer, but also HIV and AIDS and emerging health challenges like Zika, Ebola, and the coronavirus outbreaks. An American Cancer Society professor and former provost and executive vice president of the University of Texas MD Anderson Cancer Center, he has served as chair of the Pharmacology and Toxicology Department and interim dean at the Geisel School of Medicine at Dartmouth, and as chair of the Board of Scientific Counselors for the Clinical Sciences and Epidemiology at the National Cancer Institute. He is now chair of the Research Council of the ACS, overseeing research funding decisions for the ACS. We actually have some funding from that, and some of you who are funded are here. As a physician scientist and oncologist, Ethan moves his work from the bench to the bedside. An example is his role in establishing curative retinoic acid-based differentiation therapy for acute promyelocytic leukemia, APL, a treatment approved by the FDA. Dr. Dmitrovsky cloned the APL translocation product, PML-RAR alpha, and patented the genetic test for this leukemia. Actually, when he was doing that work, one of the fellows at MSK was Barbara. He received the American Society of Clinical Oncology and ACS Award and lecture for these discoveries. Dr. Dmitrovsky graduated magna cum laude from Harvard University, also a good school, and received his medical degree from Cornell University Medical College, actually happens to be my alma mater. His residency in internal medicine was at New York Hospital Memorial Sloan Kettering, and oncology fellowship was at the National Cancer Institute. On a personal note, I've been fortunate to know Ethan for the majority of my career. He worked very closely with us when I was at MD Anderson with my late mentor, Wan Ki Hong, certainly in the area of retinoids and cancer prevention. And I've been very fortunate to stay in close touch with him over the career, and he's been a friend and mentor to me and many others. So we actually have a special program today because after this round, since I know he's such a good mentor, he's going to meet with trainees of the Cancer Center. And what we've started to do this year under Dr. Dinan's leadership is it's all trainees. It doesn't matter what department you're in. Actually, if you're 50 years old and you still consider yourself a trainee, show up, and you'll get some advice. Mike Hurwitz in the back is going to bring the fellows. So we're going to do that from 1 to 2 over in the Calabrese room, 208. Unfortunately, the room in the back was booked today. So before we start, Ethan, I have a gift for you. We saved time. We already did 3 pictures, but it's the new thing. It's a wonderful globe that says Yale Cancer Center, so thank you. We can get another picture if anyone wants to do one. We might as well. You can never have too many. And Ethan, the floor is yours.
Thank you so much for it. I've never had anyone applaud before I began to speak. Roy, what a pleasure to be here. What a joy to see so many friends and colleagues from other institutions that I've worked with. Let's begin. I'll be talking about new cell cycle inhibitors that can eradicate aneuploid cancers. I have nothing to disclose except that I'm an employee of Leidos Biomedical Research. What I'm going to tell you about is the use of retinoids, natural and synthetic derivatives of vitamin A, that can be used as tools, discovery tools, to uncover previously unrecognized downstream therapeutic pathways. And these will include a pathway that my laboratory discovered that we've called anaphase catastrophe. You'll hear more about it later. But what we have uncovered is a new cancer pharmacology where we can selectively target for eradication aneuploid cancers whilst relatively sparing normal cells. And I'm going to tell you about one such story that we just published a few weeks ago involving cyclin-dependent kinase 2 inhibition, CDK2 inhibition. By way of review, retinoids are natural and synthetic derivatives of vitamin A, which is essential for vision, fertility, and immune function. Retinoids have distinct functions during development, but also play a pivotal role in cancer therapy and prevention. And as Roy said, one of the early stories of retinoids used in the clinic was the acute promyelocytic leukemia story that we played a role in. And retinoids signal through nuclear receptors. Nuclear receptors can be activated either by all-trans-retinoic acid that we were involved in FDA approval that activates the three so-called retinoic acid receptors, RAR alpha, beta, and gamma. And another drug that became FDA approved called bexarotene predominantly works through the heterodimeric partner, RXRs, alpha, beta, and gamma. So all you need to know is these two proteins come together and they signal downstream changes in gene expression that lead to the therapeutic effects that we see in the clinic. So I'm going to show you just one image from the paper we published in the New England Journal of Medicine that shows when you treat an APL patient, shown here, with retinoic acid oral therapy for just 10 days, you change dramatically the appearance of these cells. And as you can see, the morphology has changed dramatically, and in the leukemic cells, you can see the our rods present in the cytoplasm. So we were involved in this story, became FDA approved, and Ron Evans reached out to me and said, let's clone the genetic cause of this disease. So we cloned the 1517 translocation, published it in Cell, that showed that whenever you had this translocation, paradoxically, you always responded to retinoic acid therapy. And then my lab went on to patent the genetic test for this disease that's widely used to diagnose APL. But what's quite intriguing is that during this retinoic acid-based therapy, what retinoic acid did was induce the destabilization of the oncogenic protein PML or alpha. My laboratory had published in PNAS that if you engineer mice with PML or alpha, they have abnormal myelopoiesis and go on to develop leukemia. So I asked this question, how could we use this model, an early model of targeted cancer therapy, as a guide to discover new therapeutic pathways? So by way of summary, when you treat with retinoic acid, you induce differentiation in leukemic cells, you degrade the oncogenic protein PML or alpha, you promote transcriptional activation, and there's very high remission rates. And so well over 90% of patients are cured now with retinoic acid-based therapy. And that stands in marked contrast to the 20% of patients who we were able to cure with chemotherapy. So this is essentially a solved cancer problem, but there's much we can learn from APL. And so in the absence of retinoic acid, leukemogenesis is promoted, there's a differentiation block, transcription is inhibited, and there's a bleeding diathesis that can only be controlled with remission, induction by chemotherapy or retinoic acid. And so what we sought to do is to look for downstream pathways that were activated or repressed by RAR agonists or RXR agonists. And we found three pathways. One that I'm going to talk about today is targeting aneuploidy. We've also seen an immune response regulation pathway and an oncogene destabilization pathway. But today I'm going to focus just on targeting aneuploidy. So everyone in the audience is well aware that aneuploidy is a hallmark of cancer. What you may be less familiar with is that this has been recognized since 1890. And here's an image from the Huntsman paper that actually codified the notion of aneuploidy being a hallmark of cancer. And then, unbeknownst to me, because I hadn't seen this paper yet, on the right, you can see a review article that my lab published recently on aneuploidy as a target for cancer therapy, and it is remarkably reminiscent of the Huntsman image. So since 1890, the idea has been that aneuploidy was potentially a cause of cancer or a drug target. I'm going to show you that it's a drug target today. With Roy being here, I needn't tell you about the importance of lung cancer as a public health threat. You already know this, the most common cause of cancer mortality for men and women. And yet, although there's progress that's been made, there's more that needs to be done. What I'm going to tell you about is a property of lung cancers that you may be less familiar with, and that is that they're uniformly aneuploid. So that raised the question, of aneuploidy as a lung cancer hallmark. Is it a driver of lung cancer survival? Is it a driver of anti-cancer drug response? Is aneuploidy itself a lung cancer drug target? So those are the questions we set out to ask just over a decade ago. And the partial answer is that aneuploidy is a strikingly negative prognostic factor in lung cancer. In the lower panel is a study that we haven't published using the Cancer Genome Atlas. What we found is that high aneuploid tumors are extremely poor prognostic tumors. So people with highly aneuploid lung cancer survive much shorter than those without. And interestingly, others have seen the same. It's a result that's frequently noted in the literature. You're aware that genetic mutation oftentimes promotes response to immune checkpoint blockade, but aneuploidy confers resistance. So the presence of aneuploidy is a negative prognostic factor, and it is a cause of drug resistance. So let me tell you how we found a novel pathway, a death program that I mentioned called anaphase catastrophe. This is a paper we published a number of years ago in PNAS, where we took benign lung epithelial cells, and in the absence of carcinogen, these cells remained benign. If you took benign lung cells and added the carcinogens that cause lung cancer, cigarette smoke, condensate, or the nitrosamine N and K, we could essentially make a lung cancer. So the question that we asked at the time is, if you add the carcinogen but add the chemopreventive agent, retinoic acid, would the benign cells stay benign or would they become malignant? And they stayed benign. And so we set out over a number of years to understand the basis for that cancer prevention mechanism. And what we found was that retinoids and rexinoids, the drugs that I told you about that activate the RAR or RXR pathway, induced the destabilization of G1 cyclin, cyclin D1, cyclin E. And we never expected that that would be the result that came to the fore. But we found that this destabilization was distinct from the repression that many of you study the EGFR tyrosine kinase inhibitors, the repression of the same cyclins, suggesting they might come together in a cooperative measure. So here's the only slide I'm going to tell you about another aspect of my laboratory. We spent about a decade figuring out how that destabilization occurs. And what we found was that retinoids will induce the well-known ubiquitination destabilization program through the proteasome, but we uncovered an alternative destruction pathway that involves a ubiquitin-like pathway that involves another small molecule moiety that targets proteins called ISG15. And in this mix, we've highlighted a particular enzyme called USP18, the D-ubiquitinase, that we feel is a very attractive drug target, and that's all I'm going to talk about in that work. So back to how we uncovered aneuploidy as being really an important target in lung cancer, we conducted 5 clinical trials over a number of years in lung cancer patients, both phase 0, phase 1, and phase 2 trials, combining rexinoids with EGFR TKIs. Remember, I said they cooperate in vitro. And what we found was that these tumors in post-treatment versus pre-treatment tumors could dramatically repress EGFR expression, repress proliferation as assessed by Ki67 expression, and then also repress G1 cyclins, both cyclin D1 and cyclin E. We found intriguingly that this therapy was very well tolerated, oral therapy, and there was a survival advantage in these patients. We went on to conduct a larger study, and we found something also interesting, which is that in addition to the survival advantage, responses were seen even in KRAS mutant lung cancers, which, as Roy and others know, is a sine qua non of drug-resistant lung cancers. So based on this work, my friends at the time at MD Anderson, and still friends, were running the battle trial that Roy played a leadership role, and they actually independently confirmed the results that I've just told you about in a second institution. So 6 clinical trials, all showing the same results. I'm a physician scientist, and I was interested in understanding the question, so how does this occur? And as I said to you, when you treat with bexarotene, the orexinoid, you repress G1 cyclins. And we asked the question, what happens if you treat with a cyclin-dependent kinase 2 inhibitor, CDK2, when you drive the kinase activity down? Would that replicate the results that we see with bexarotene, or would it lead to a different phenotype? And the first thing that we did is we engineered a mouse, published this in PNAS, that overexpressed cyclin E or a proteasome-dependent cyclin E species. And what we found was that lung cancers arose. They were all adenocarcinomas. But if you destabilize cyclins, you develop metastatic cancer, unlike the normal protein, and you develop multiple lung cancers. So this is the first clue that we had that aneuploid was important, because if you examine these tumors by FISH analysis, they're all highly aneuploid. So cyclin E overexpression is driving an aneuploid tumor. And we asked what happens when you inhibit this pathway. And so we began a decade-long study to target aneuploid lung cancers and other cancers with specific CDK2 antagonists. And this effort led us to this novel death program that I've referred to called anaphase catastrophe. So here in the upper panel, you'll see the drugs that we have looked at. Here are some representative publications from my lab. But now let's focus in on this lung cancer cell that is going to develop 3 progeny. And you'll only see 2 because one of the cells will fall out of the focal plane. So it goes through this bizarre division. So there's not death in mitosis. The progeny cells survive. And now you can see that the 2 progeny cells, after mitosis, involute and die. So that's what anaphase catastrophe is. It's not death in mitosis, it's death of the progeny. And so we were fascinated by this result and began an effort to understand in greater detail how this death program occurs. And the first fascinating observations that we made was using a robotic screen that we did with Jeff Suttleman a number of years ago that was linked to the Sanger genetic database. And we found unexpectedly, and perhaps even paradoxically, that CDK2 inhibitors would repress growth of all lung cancers, but were exquisitely sensitive to those lung cancers that harbored a KRAS mutation. That's contrary to the dogma. So we had to answer a question: how is it that a CDK2 inhibitor preferentially leads to responses in KRAS mutant tumors? Okay? And over several years, we identified over 20 potential candidate mediators of this effect. In the upper panel, what you can see is we looked at aneuploid lung cancers that harbored a KRAS mutation, and we were looking for what we called factor X at the time, which would predispose to CDK2 inhibitor response and control anaphase catastrophe. And we discovered that factor X was a centrosome protein called CP110. CP110 is a direct phosphorylation target of cyclin-dependent kinase 2. It has 10 phosphorylation sites. We mutated each of those 10 phosphorylation sites individually, and we found 2 that controlled anaphase catastrophe. So tuck this thought away in the back of your mind, because I'm going to show you some data. There's an 11th phosphorylation site. That 11th site is not CDK2 dependent, but it's dependent on another kinase called polo-like kinase 4. Okay? 10 phosphorylation sites, only 2, when you mutated CP110, abrogated centrosome clustering and abrogated anaphase catastrophe. So we put those mutated CP110 variants back into lung cancer cells, and here's what we found. We found that of these 2 sites, only 1 was the dominant mediator of anaphase catastrophe, but that mediator prevented clustering of centrosomes and prevented downstream signaling of anaphase catastrophe. And that's what this result shows, is that effective clustering of centrosomes in the bipolar anaphase state was seen, but in the multipolar state, we didn't see this, and death was the result. And you might be wondering, is this a general property of aneuploid cancers, or is this specific to lung cancer? It's a general property. So in subsequent work, here's some representative data from colon and pancreatic cancers. We've actually seen this effect in every aneuploid cancer model that we've looked at. What we found was that, once again, colon and pancreatic cancers are responsive to CDK2 inhibition, but they're particularly responsive when you have KRAS mutations. And so this led us to wonder, how precisely does CP110 act in this way? So I'm going to stop and just review what I've just said to you. What I've told you, there's a new death program, unlike mitotic catastrophe, that's death of progeny cells. This is occurring specifically at the anaphase of the cell cycle. The death program does not depend on synthesis or destruction of centrosomes, but depends on centrosomes that are there that don't cluster anymore. Okay? There's a specific protein called CP110. That's a centrosome protein, and tuck away the idea, is there some centrosome abnormality that occurs? That is sensitizing to KRAS mutant lung cancers. How does that occur? Is CP110 involved? So we solved that question, and I'm just giving you a summary. What we found was the reason that CP110 sensitizes in KRAS mutant lung cancers is that KRAS is triggering, through a cyclin F-dependent mechanism, the proteasome-dependent destabilization of CP110. When CP110 falls below a critical level, then you will induce the regulation of the death program. And is this translationally relevant? We actually developed a paraffin-embedded histopathology, published this in Cancer Research, histopathology analysis of CP110 in 550 lung cancers. And we found that the same regulation that I've shown you in vitro is actually seen in human lung cancers. This relationship actually translates into the clinic. So for those of you interested in how this might be clinically relevant, what I've just told you is there is a phosphorylation target, CP110, that seems to track with anti-tumor response with CDK2 inhibitors. And so we've made antibodies to those 2 phosphorylation sites that were phosphospecific, only one of which is serine 3170, is the dominant mediator of anaphase catastrophe. We also made an antibody to the less active site, and then we made a third antibody to a kinase phosphorylation site of CP110, separate from CDK2 inhibition. And in the left-hand upper panel, what this shows is that when you treat with a variety of CDK2 inhibitors, you track a repression of the phosphorylation state during the course of the treatment. So the cells will reduce phosphorylation with a CDK2 inhibitor. It's just what you would expect. But what about tumors. And so we've also done this experiment with xenograft models. And what we've been able to show is that when you treat PDX tumor or a xenograft model of a cell line and treat with a CDK2 inhibitor, this assay actually monitors treatment response. So, Roy, this is a potential companion diagnostic. One could envision that circulating tumor cells you could assay with these phosphospecific antibodies and see whether the patient's tumor is responding. So, so let's go a little bit more deeply into mechanisms of action. And so we have been studying in detail the mechanism associated with this death program. And we've been using as tools live cell imaging and a technology called focused ion beam scanning electron microscopy. So I'll discuss these. And what you'll take away from these experiments is that we have a striking therapeutic window between aneuploid cancers and normal human alveolar cells, favoring the death of the cancer cells and survival of the normal cells. So let me show you what focused ion beam scanning electron microscopy is first, and then I'll show you the results. So this is a way to essentially obtain analogous to a CT scan of a cell. This is how we prepare samples. Tissues or cells of interest are placed into a resin mold. On top of the resin, so the resin extracts the water. On top of the resin, we embed with a scoring system so we know exactly where our chevron scoring. And then a platinum pad is deposited over the sample. That's really to secure the orientation so we know exactly where the tissue is. And then we mill the sample, exposing the cell of interest, and perform scanning electron microscopy on each of the images. And because they're stacked, we can use computational image analysis to determine three-dimensional structure by overlaying the individual milled stack and get a three-dimensional image of the organelle or cell of interest. And so here's an example where you can actually take a single image stack and then develop a three-dimensional model of the tissue organelle of interest. So we've used this technology to look at centrosome structure. Remember I said CP110 is a centrosome protein. And you remember that centrosomes are composed of two centrioles. So let's look at the three-dimensional structure on the left of a bipolar division of a lung cancer cell. You can see, if you look closely, two centrioles. But now let's look at the multipolar mitotic event. Is there a single centriole or is it double? It's a single. Look closely, you'll see that the tripolar cell has lost its centriole partner. And this is really a likely explanation for why this death program occurs, because the cells can't get through the division unless they have some way of surviving. So in the lower portion of the slide, it shows that mononuclear cells are the product of bipolar division, largely with CDK2 inhibition. But what we've discovered is that aneuploid cancer cells, unlike normal cells or bipolar division, have multiple cell fates. And those multiple cell fates each have their own biology. And that is a potentially clinically tractable biology. And I'll discuss that in a little bit. To confirm these results, we obviously used immunofluorescence, but since we've just published this work, I'm not going to discuss this. So I've planted the notion in your mind's eye that looking at the progeny fate is a very important aspect to understand why CDK2 inhibitors work in the clinic or why they may not work. So in order to study cell fates, we introduced 4 Fuji probes into a variety of lung cancer cells. Each Fuji probe fluoresces at a different phase of the cell cycle. And that allows us to determine if they die, when they die in the cell cycle. And because we use live cell imaging, we can actually follow these colored cells of different colors telling us where they are in the cell cycle. And let's take a look in the left panel, what happens to that dividing green cell. So it goes through a multipolar division, 3 progeny. What happens to the progeny? They all die. That's anaphase catastrophe. That's the second time I've shown it to you. Everyone with me? But there's another fate that occurs that I think is really clinically relevant. Let's look at the right-hand panel. Here's a cell that goes this very strange division, but notice it's become multinuclear. Multinuclear. And that immediately had us begin a path to understand what is the nature of the multinuclear cells. That's what I'm going to talk about now. So here is a diagram that tells you the summary of cell fates of aneuploid cells treated with CDK2 inhibition. Some cells go through bipolar mitosis, about a third. About a third go through multipolar mitosis, and about a third immediately die through apoptosis. But those cells evolve under continuous CDK2 inhibition. They can evolve into double nucleated cells, failed cytokinesis, some of which will die through apoptosis. But one of the unexpected phenotypes that we discovered was the presence of multinuclear cells. Multinuclear cells. Is that of any importance clinically? Is it of any importance biologically? Well, the answer to that is yes. So we then began to study the properties of these different fated cells. And what we found was that polyploid lung cancers were resistant to apoptosis induced by CDK2 inhibition. And this turns out to be quite relevant clinically. Why is it relevant clinically? So what we have found is that CDK2 inhibition, we in the field has found, can lead to dramatic and statistically significant repression of tumor growth in PDX models. But there's always a population of persistent tumor cells. What is the nature of those persistent tumor cells, and can you eradicate them? So I've already told you that our suspicion was that these are these polynucleated cells. And so if you look at the left-hand panel, you'll see we found early on these persistent cells had one feature in common. These are all PDX tumors. They all had residual polynucleated cells. And they all persisted. And so can you actually see the evolution in vivo of these polynucleated cells? So we use a technology that you're likely familiar with called intravital lung cancer imaging, which we transplanted the 4 Fuji probe transfected cells into recipient mice and then imaged the living mouse harboring a lung cancer cell. And in panel B, what that shows is over the course of treatment in every mouse, there was an accumulation of these polynucleated cells. What is the biology of the polynucleated cells? So to elucidate that question and to address it, what we decided to do is purify populations of lung cancer cells, those that were part of the non-aneuploid population and those that were part of the aneuploid population. And then we conducted RNA-seq analysis to determine, was there differential gene expression in the aneuploid population vis-a-vis the non-aneuploid population. And what we found was fascinating to us, and I hope of interest to you. What we found is there was an unexpected enrichment in the aneuploid polyploid population of CDK1 and KIF family members. And this enrichment was not an enrichment that we expected at all. But it led to yet another question, which was, is in the persistent polyploid cancer cell populations that preferentially overexpress CDK1 and KIF family members, is that linked to the aneuploid state? Does it affect lung cancer survival? And obviously, will targeting this specific fated population, will that eliminate the cancers? And so now I wanted to remind you that what we've talked about are multiple fates. And the idea that I'd like to leave you with today is that each fate has a different biology and perhaps a different clinical biology. Let me make a comparison. We're all familiar with EMT, epithelial to mesenchymal transition, and we all know that there are dramatic changes in drug sensitivity. This is a little bit akin to that. So let's now take a look. So using the Cancer Genome Atlas, we analyzed representative mediators of this persistent population, or presumed mediators, CDK1, pol kinase 1, or kinase A, and multiple KIF family members. What we found was that if you overexpress these species, you were always an aneuploid tumor. But if you were not overexpressing and had normal levels, you were far less likely to be aneuploid. But what about the actual clinical biology? So what's the prognosis? And as you can see, comparing overexpression of these different persistent population cells, there's a dramatic effect on prognosis. So if you have overexpression of these species, then your survival using the Cancer Genome Atlas as the tool is markedly reduced. So this led us to this question, which is, will combined anti-cancer therapy with CDK2 inhibitors plus KIF1 or CDK2 inhibitors plus CDK1, KIF1, will they eliminate these cancer cells? And here's the results. So we combined CDK2 with a variety of KIF inhibitors or with CDK1 inhibitors. And what we found was, using subtherapeutic dosages to tease out this effect, we saw remarkable cooperation with these two agents and led to the loss of these persistent cells. So, Roy, that's a clinical trial worth considering, right? And that would be something that would allow us to do something that we haven't seen yet with CDK2 inhibitors, which is to repress tumor growth, but also to eradicate tumor growth. So this seems to be true with all the CDK2 inhibitors. Here are the CDK2 inhibitors that are now in clinical trials. And not one of them is considering this difficulty of how to address persistent population of cancer cells. And so my urge recommendation to my clinical trial colleagues is consider why we don't eradicate tumor cells with a CDK2 inhibitor. And it's a little bit reminiscent when the first drug in class moves into the clinic and the obvious experiments are done, and they may lead to stable disease, but not involution of tumors. And then the drug gets dropped. We really have to have a an approach to try to eradicate this population. So today, in the interest of time, I've only had a chance to tell you about one class of drugs, CDK2 inhibitors, but actually that's not the only class of drugs that we've been working on. Turns out there's a new cancer pharmacology, and coincidentally, Tak Mak, who is at Princess Margaret University of Toronto, called me up because he and I have been publishing the same observations, but he was publishing using PLK4 inhibitors and threonine tyrosine kinase inhibitors. So Tak Mak and I have joined forces, and we published a series of papers. I'm just going to summarize them here, where we've now demarcated multiple sites of induced death of aneuploid cancers with polo-like kinase 4 inhibitors, CDK2 inhibitors, and TTK inhibitors. These are reasonably well tolerated in the clinic. They're all in phase 1 or phase 2 trials, and they can be added together. Another thought for the clinical trialists in the room. So what I've told you today is that aneuploid is a hallmark of cancer. It's a hallmark of lung cancer. And I put forth the proposal that this is a tractable target to eradicate aneuploid cancers using a combination or individually of these 3 drugs that I've just told you about. These each engage distinct mechanism of action. Only the CDK2 inhibitors induce anaphase catastrophe. Other death programs are induced by PLK4 inhibitors and the TTK inhibitors. And since all of these drugs are moving through clinical trials, the work that I've told you about today is translationally relevant. And here's a photo of my happy lab, and they are truly happy. I asked them to smile. And before I end, I always leave a few minutes to tell you a little bit about the institution where I work at, Frederick National Lab, because most people are not familiar with this ecosystem of national labs. They exist, they're federally mandated labs that exist to tackle problems that can't be addressed in academic centers or in the biotechnology sphere. So Frederick National Lab was created just over 50 years ago when President Nixon came to Fort Detrick, Maryland, to the base at Fort Detrick, and he did something of historic note. He decommissioned, he demilitarized 70 acres of what was then biowarfare facilities. And he took those weapons of war and repurposed them for a peaceful end. The peaceful end was to create what we call now the Frederick National Laboratory for Cancer Research. 10 years ago, when Harold Varmus was NCI director, he gave us a challenge, which was to launch the RAS initiative that I suspect many of you have heard of, the Frank McCormick and Dwight Nisley direct. And the purpose was to understand the intricacies of RAS signaling to create new drugs to inhibit RAS. I'll talk about that in a minute. One of their early successes in our history was that we facilitated with the National Institute of Allergy and Infectious Disease the first successful blood test to secure the safety of the American blood supply from the HIV contamination. So overnight, contamination through blood product use stopped. We also support a number of trials worldwide in resource-constrained countries. We have worked with Mpox in the Democratic Republic of Congo, Ebola outbreak in sub-Saharan Africa, and we played an instrumental role in the coronavirus outbreak. To give you an example of what I mean of doing work that can't be done elsewhere, raise your hand if, like me, you've taken a nasal swab for the coronavirus. So did you know that every one of those tests was validated for the FDA at the Frederick National Lab? And the reason for that is that every sample that we were given, we had no idea who the vendor was. They were all de-identified. But the vendors would give us the kits to test because they knew that we were a safe harbor. And so what we did is we repurposed our HPV program to create what you've probably heard about, the SeroNet initiative, which has been doing all of the serologic studies in the coronavirus. So that's an example of what we did. Another example is that Doug Lowy, early in my tenure when he was NCI director and still is, he asked me to set up as a national initiative a cryo-M facility. So we have 4 cryo-M machines exclusively devoted to extramural cancer scientists like yourselves. And we provide image analysis at no cost to the scientists. I mentioned to you that we work in challenging settings. We have about 300 clinical trials in our portfolio. And I was trying to find one example to show you what I mean by doing work that can't be done elsewhere. So I'm going to show you an image from Liberian national TV. The Ebola outbreak first occurred in Liberia. We were on the ground, and we facilitated a 4-arm clinical trial for Ebola, where 2 drugs reduced mortality from about 90% to about 12%. One of those drugs we manufactured, and they both became FDA approved. So in Liberia, we have built the C.H. Rennie Hospital Research Wing. And that houses these precious, irreproducible Ebola samples and now coronavirus. And these settings are very challenging because many of our clinics have been attacked by militia, and I've had to extract our clinical teams. And I think most deans of medical schools probably wouldn't send their staff to those conditions, but that's where we work. So here there was a fire in the research wing, and watch what happened. So the people that you see in this bucket brigade are actually our employees, and they put the fire out by saving our research wing. But because we were familiar with the risk of arson and also of military activities against Westerners, we had used wire cutter resistant cable to secure all of our equipment that housed these precious samples. And we had walls and doorways that no one could enter. And so not a single sample was lost. And in the course of 2 weeks, the research wing was up and working. That's the kind of work that we do. Another example is shown on the right, which is a map of the Democratic Republic of Congo. Do you see that red arrow? That's the city of Goma, where we have a team deployed. You probably heard there was a volcanic eruption in the Democratic Republic of Congo. Well, it turns out it was exactly in that city of Goma, and I had to extract our team to safety. And they all volunteered to come back. And did you hear about, in the news just a few weeks ago, the M23 militia attack? It was in the city of Goma. So I had to extract them again. And they're now working at a distance in Uganda. Do you see that black arrow? That was in the news recently. That's the city of Kindu. So remember, we're running complex clinical trials just like you would run here, but under the most difficult circumstances. And so we have to preserve continuous supplies of cold storage of our therapeutics at every step of the way. And the city of Kindu is where our staging area is still. And militia attacked that city, and we had to shelter in place, but fortunately they were safe, and we've been able to keep working. And to show you evidence of that, while all this was underway, our NIAID colleagues with us published a paper in the New England Journal of Medicine from the trial that we were running for Mpox for tocophermat. And so we're proud of this public service that we do, and a good example is that the first 2 drugs for coronavirus, we facilitated those trials, worldwide 70 sites that led to the FDA approval of remdesivir and baricitinib. We also manufactured the vaccine for Zika that was used in clinical trials mainly in South America. And I just wanted to end by showing you that after over a decade of work that Harold Varmus had encouraged us to begin, we now have 3 drugs that are RAS inhibitors in the clinic undergoing clinical trial testing. These are either best in class or first in class. We received fast track status for our inhibitor that works in RAS in the on and off states, which is a best in class feature. We have other drugs that are called RAS breakers that are in clinic, and we have a pan inhibitor that just moved into the clinic. So let me end by saying thank you for inviting me to visit. And if you're interested in learning more, we actually had 2 papers in Science in the same issue, and a third was a commentary. Here's the commentary. So I want to thank my collaborators and thank you for your attention.
Thanks, Ethan, for a wonderful tour de force there, and I'm thrilled that you're working on lung cancer. Actually, it reminded me you were a key component of the battle trial, and that was arm 4 of the battle. Now I know where it came from. I sort of remembered that. So we have time for a few questions. I have a few online, but I'm happy to take the in-person ones first. Anyone have a question? Megan? Here, I'll bring you the microphone. It's my job here.
You're doing a great job, Roy.
I was curious what, once the cells become a polyploid,
Yeah.
will they continue to divide, and can they ever revert or can they ever revert out of that polyploid state that you've
Yes,
observed?
yes, they do. Some die, but most continue to divide. And that's what we published just a few weeks ago in JCI Insight, if you want to take a look. And they're the source of the persistent population.
We have a question from Timothy Robinson online. Can you comment on what we know about how aneuploidy inhibits immune recognition? Is there a way to specifically enhance immune or cellular therapy in aneuploid tumors?
I wish I knew the answer to that excellent question. We have not investigated that. What we have investigated, remember I said there were 3 pathways that we've been studying. One is chromosome instability, aneuploidy. The other was this destabilization pathway. The third is how retinoids affect immune function. And so we've been pretty active in that area, but we've not asked that question, which is a really important question to ask.
I'll take the priority and ask a question since I'm the moderator. In lung cancer, EGFR mutant lung cancer, we've made so much progress there, but, we still have persister cells. Many of the drugs are cytostatic and not cytotoxic. The biggest advance we'll hear in Barcelona next week at the World Lung Meeting is adding chemotherapy back to targeted therapy. And I'm not sure that some of these VEGF targets are going to do it either. With the approaches that you mentioned here today, are those applicable to EGFR mutant cells? Have you looked at cell lines?
Absolutely. And I would actually encourage those who are interested in that question to follow up. And it's been a really powerful tool. Megan and I were talking about looking at cell fates and kind of new to this area, but it's been an incredibly powerful tool to understand what is the population that persists after, say, an EGFR TKI treatment. I think that's really relevant.
We have a good showing from the Paletti lab here. They're taking notes, I hope. Okay, David.
A quick one, kind of a follow-up to Megan's. What is the fate of those multiple nuclei? Do they eventually fuse so you end up with a single polyploid nucleus?
I didn't talk about that. We were really interested in answering that question, so we just published that these are not single fused nuclei. Each one is distinct, and we used the focused ion beam scanning electron microscopy technology to confirm that. And we also used laminin staining to, so that's a separate question. Great, so that's a separate question. So there's no uniform fate. Sometimes they will die, sometimes they'll stop growing, but mostly they recycle. And then when they recycle, they could go into any of the progeny that I talked about. So it's a continuous recycling. And that's why this notion of combination therapy to find what would preferentially eradicate the persistent population is so appealing. Although we haven't shown this, we suspect, hypothesize, that'll shut down the evolution. And then they will die.
Barbara?
Wonderful talk, Ethan.
Nice to see you again.
Nice to see you. Many of the things that seem to be hallmarks of these aneuploid populations strike me as also recognized hallmarks of EMT.
Yes,
And particularly as you think about
Yeah.
downstream from Egypt.
which is
All right.
why I made that analogy.
So I'm wondering sort of which is the chicken and which is the egg, and can we apply what we know about EMT to these aneuploid cell death pathways?
Yeah, we've been reading a lot about, we don't study EMT in my lab, but we've been reading a lot for exactly the reasons that you said. We feel this approach of following the fates of cells with multiple Fuji probed vectors is a great way to interrogate the question that you're asking. This transition state is a distinct transition state from EMT, but it's quite reminiscent of going from a drug sensitive to a resistant state. And as the prior questioner asked, the complexity here is that the cells seem to recycle. So they're always enriching the tumor. And if you recall, I showed you the PDX analysis of the persistent tumors. There's an abundance of these polynuclear cells there. So they're obviously occurring at least in a PDX model. We haven't yet looked, Barbara, in vivo, in human tumors.
Okay, last question. I'm going to have Wei pass you the microphone.
Thank you. So these cells you describe going to an apoptotic death afterward. Do you have any idea what is actually the trigger for the apoptotic death, and whether it's the centrosome abnormality or genomic instability or something else that happens?
What we've seen as a general, remember I said that with TechMac, we've been studying this new cancer pharmacology of targeting aneuploid cells. It seems to me that in all of these death programs, there's a critical threshold where the cells can't survive if there's too much chromosome instability. So we're seeing that as a common thread across PLK4, TTK, and CDK2. The question you're asking is a fascinating one for a lot of reasons, and I'll be very brief in saying is that apoptosis is not the only death program that can occur. And so we're actually looking very carefully at all the possible death programs that are conferred by this anaphase catastrophe. And we'll have an answer pretty soon. We're really kind of open-minded about what could be the cause.
Great. I think we'll have to stop now, but anyone who has questions can join Ethan now in WW208. I see our chief fellow here from hematology oncology and Michaela.